start. So today it's just for me to have here Rudolf Toman. So we're working with Rudolf since around a year now, because he's one of the guys who will produce the LPS from Coxiella Burnetti. It's one of these elements which is necessary for us to understand the physiopathology of the Q flavor and the way for Coxiella to avoid the destruction by phagosers and by macrophages. So Rudolf is working in Slovakia. He has this group in Slovak Academy of Science, so for diagnosis and prevention of lipopolysaccharide and chemical infections of the past war. And today we'll talk more about biochemistry and the structure of the LPS of Coxiella Burnetti compared to the other LPS. So thank you, Rudolf.
Thank you. Can you hear me?
Yes.
Okay. Thank you for your kind interaction. So I'm really happy to be here again in Marseille and to discuss our ongoing research on Coxiella Burnetti. So actually we have been working with this lab here for more than maybe 15 years, something like that, for a long time, and now still I came over again to discuss future work on this very interesting pathogen. So today I'm going to talk about the structural and functional peculiarities of the lipopolysaccharide of Coxiella Burnetti. And first I would like to tell you why to study lipopolysaccharides of gram-negative bacteria. The first reason is because these LPS are the major determinants of virulent expression infection of many gram-negative bacteria. They represent the dominant surface antigen and are targets for the host immune system. They may act as protective immunogen, and they may contain unique structure motifs, enabling rapid and highly specific detection and identification of this bacteria. And this is also particularly of huge interest for Coxiella Burnetti, because as I'll show you later, contains two unique sugars. They are highly specific and they are found only in Coxiella Burnetti, and that's why those two sugars are very important biomarkers of bacteria. And therefore, the structure-function relationship studies of this LPS are of potential interest, not only from structural point of view, but also from the biological properties. So for those who are not working too much with LPS, I'll give some basic information on LPS, on what's also called endotoxin. As you can see, LPS in gram-negative bacteria is located in the outer membrane of these cells of gram-negative bacteria. It's huge, usually huge, bulky, post-specific chains, and then lipid A portion, and this lipid A portion actually is anchored to the surface of the outer membrane. As you can see, this post-specific chain of LPS is exposed actually to the immune system, and these host pathogen interactions are therefore very important, and structure-function relationship studies are also very important. So also there's something also for those who are not working with LPS. I should say here that LPS, typical LPS, is composed of three major regions. The first one is the lipid A, that is actually responsible, it's quite simple, but it's responsible for endotoxin activity of LPS. This lipid A is usually very cryptic, so it's very similar in many gram-negative bacteria, but it's quite different in Coxiella Burnetti. Then we have a core region. Again, this core region is also quite cryptic, so it's very similar in many gram-negative bacteria. Then you have inner core and outer core. This outer core is much more flexible. And then you have an O-specific chain, and this O-specific chain has usually is composed of repeating saccharide units, but it's a highly variable region. This is the most variable region of LPS, and it's changing during switch-on, switch-off mechanism, depending on the environment in which bacteria this present. So there's some basic information for those that have worked with Coxiella Burnetti. Very briefly, is a gram-negative obligate intracellular parasitic bacterium, belongs to the gamma subgroup of proteobacteria of the legionnaires, Coxiella, genus Coxiella. Coxiella Burnetti lives and multiplies. It's not completely true. It's not completely true now, according to new findings, but it's officially still written right there. That's why this studies on multiplication of Coxiella Burnetti of utmost importance, and new findings that have been done here in Marseille are very important. Explain all these stories here. And then, of course, Coxiella Burnetti causes coccidiosis in animals, which is known as Q fever in humans. You know, there was a huge Q fever outbreak in the Netherlands maybe 2 years ago, and that's why now Q fever and Coxiella Burnetti is a subject of very intensive research studies. This information is also well known for those who are working in the Coxiella Burnetti field, that in nature occurring Coxiella Burnetti cells in virulent phase 1 change to virulent phase 2 cells upon serial passages in embryonated eggs. And this phase variation is connected with modification in both composition and structure of Coxiella Burnetti LP lipopolysaccharides, proteins, and also phospholipids that we've already shown. But this phenomenon of phase variation is still poorly understood. We can see here this phase variation very nicely on a very simple 1D SDS page. Here you see it's a LPS lipopolysaccharide isolated from virulent phase 1 cells. It's called we call it phase 1, and then you see LPS isolated from virulent phase 2 cells. So you see at first glance that there was dramatic change in both in composition and structure of most of those LPS. And here you see LPS of this is from Salmonella typhimurium. And you see here that has some typical ladder-like bending pattern, which is typical of LPS with regular or specific chains. So with this repeating, this you cannot see here in Coxiella Burnetti LPS. It's highly homogeneous. And that's why the studies on structure of this LPS are quite difficult. So we did in the past structure work on this LPS too, and this was not so difficult. So we were able to show that this lipid A portion, which is very tricky here in Coxiella Burnetti, I'll talk about it later, but here is a typical to lipid A. This structure here is bound to lipidase to KDO, it's a 3-deoxymonooxidase acid. We have structural arrangement here. These KDOs are linked by glycosidic bonds. Then we have two heptoses here, and to this heptosis, terminal mannose is linked to position 3 and to position 4 of this. So that's what's not so difficult. But what were early suggestions on phase variation of Coxiella Burnetti was said this Coxiella Burnetti phase variation resembles to SR variation of complicated bacteria. And it was thought that it's proceeding like that during passage in embryonic eggs or tissue culture from phase 1 to phase 2. Partial degradation, continuous degradation of this LPS occurred in this way, like this. This low specific chain is degraded here, and then at the end you end up, with deep rough LPS2 what we had. I've shown it on the previous slide. That was a story. What's the difference between Coxiella Burnetti phase variation and first phase? That's irreversible process compared to SR phase variation that's based on switch-on mechanism. So this is actually reversible compared to Coxiella Burnetti, which is really irreversible. So when they looked on Hoover et al., Americans, they looked at what happened during this phase variation, and they were able to show that there were huge dramatic dilation on chromosomal DNA regions. Here is for NIMA phase 2, isolate strain, and so you see there were huge dilation maybe in the genes. They were actually responsible for biosynthesis of all polysaccharide chain. But they were able to show that there was another strain, isolate RSA 514, and this deletion were even larger than they've shown. They were even larger than for the NIMA phase 2 deletions. But when we started to investigate this RSA 514 strain, that it's also called crazy because it has some strange, crazy behavior. So we found on SBS page that this is a LPS from phase 2, from everyone phase 2, and this is from LPS 2 from this crazy string, that apparently that not all genes encoding all polysaccharide biosynthesis were related. So you see here, actually here, that's a region where there's all specific chain starts here. And when we did some composition analysis, so we were able to show that LPS 1 contains here this in vivo phase of virinals, mannose, peptose. I'll talk about these two sugars later. And then you have DDHTOs, galactosamine, and so on, and lipid. But this lipid LPS from crazy doesn't contain virinals, but contains all the other sugars. And when we look at this LPS 2, which is from everyone that has these deletions, so we see it's only two sugars that are mannose and DDHTOs. And lipid is the same because lipid is triptychic, so it doesn't change through this phase variation at all. So what are these major discrepancies here? The first one is that the extent of chromosomal DNA deletion doesn't correspond to both composition and structure of the LPS molecule. That's the first one. And also, Masson from the United States and this group has shown that the existence of several phase 2 isolate, what he actually claims, is no apparent relation to LPS biosynthetic region. That's the present take. So what we did in the past, when we isolated LPS from virulent phase 1, and then when we continued with isolate LPS during this phase variation, let's say from X 60 and then from X 90, so we saw very interesting things. Then here we have X 3 here. We had LPS, it's from virulence, this complete chain. At the beginning of this phase variation, of the beginning of the cultivation in virulent X. Then we had a little bit intermediate phase, and there was very little phase 2 already there. But when we continued with this growing the cells and this proceeded this phase variation, we saw a dramatic shift from this LPS redistribution of cell population. We had a very little of this phase 1 LPS, and there was a huge peak for LPS 2. And then we came in theory that we have actually all this when we have a virulent phase 1 cells at the beginning. So we have always some kind of mixed cell population because we always have some cells in which this polysaccharide synthetic region there's a defect on. Some cells have a defect in LPS biosynthesis, and then we have some cells with defect in LPS biosynthesis. They have deleted these genes for biosynthesis of LPS genes. So you see it here. So in blue, these are phase 1 cells. This complete LPS, and you have phase intermediate cells, green. They have a truncated LPS, and then you have a phase 2 cells in red. And during this phase variation, actually, because there is a need in in embryonic hen eggs, we have immunoincompetent host. So there is no need for this bacteria to biosynthesize the whole cell, I mean whole LPS, or polysaccharide chain, because it's a huge energy expenditure. And so the cells, they produce or modified or truncated LPS, LPS preference. So you have it here, it's again, you have less cells in phase 1 and more cells in phase 2, and at the end you end up with the cells in phase, what we call phase 2, you have a prevailing amount of cells, they biosynthesize only truncated LPS, these are red cells here. Then you have some cells, they have a truncated LPS, and very few cells is complete LPS. So what are the major assumptions here? That the strains or isolate contain cell population that express multiple LPS structures. That's the first postulate. And that's what I already said, that during the serial passage in embryonic eggs, in Coxiella Burnetti, redistribution of the existing LPS population takes place. And due to the increasing prevalence of those cells in the whole cell population that express LPS molecule, those that have a truncated those specific chain, and those being of R-type. So that was our conclusion, that the cells that synthesize an incomplete LPS molecule have a growth advantage during the Coxiella Burnetti cultivation in immunologically incompetent hosts. Next is what is the impact of phase variation of Coxiella Burnetti. So this is a well-known fact that has been known for maybe 30 more years, that in acute Q fever, the antibodies against phase 2 corpuscular antigens are detected earlier and at much higher titers than those against the phase 1 antigen. And again, in chronic or persistent infection, the titers of phase 1 antibodies similar or higher than those of phase 2 antibodies. And this phenomenon was also investigated in more detail, and it was found that the phase 2 antibodies are directed again, mostly again, proteins on the outer membrane of Coxiella Burnetti, and in fact, many immunoreactive proteins have already been detected, maybe 150, 200 immunoreactive proteins, but only some of them are typical for Coxiella Burnetti, and already here some of them were already actually used as a candidate molecules for detecting acute and Q fever. But still a lot of work has, in my opinion, has to be done to find really or to prepare recombinant proteins that they could be used in a detection of Q fever, because some of these recombinant proteins, when the natural proteins were immunoreactive, but when recombinant proteins were they lost their immunogenicity. Well, and then this phase 2 antibodies, we showed that these phase 2 antibodies are preferentially bind to epitope with virinals, dihydroxystreptose. And as I showed that these two sugars are unique for Coxiella Burnetti, they are not found in any other gram-negative bacteria, and those two sugars actually are, I would say, immunodiagnostic markers and biomarkers for Coxiella Burnetti. So we found it in this case, when we selectively remove those two sugars, virinals and the dihydroxystreptose for this specific chain, the serological activity has dropped considerably. So we were able to show this in this way that the sugars are really involved in phase 1 antibody binding. We then isolated both sugars. We were interested to know these sugars too. And we isolate those sugars from Coxiella Burnetti, and we showed here there's a more or less structural chemistry. And we were able to show that the first sugar is a 6-deoxy 3-symmetric glucose virinose, and has this C1 conformation, and this fibrinose, and the other one was shown to be dihydroxystreptose, was shown to be furanose. But when we isolated for NMR studies, this furanose and dihydroxystreptose switched to more stable pyranose form. But as I said, very unique sugars, very good carbon molecules for rapid detection of Coxiella Burnetti. And also we have some, and we assume that those two sugars could be involved in signal transduction pathways. So we are working on this area. Ego and general measure on this problem. So when we look more closely to this LPS, we're able to show this, as I said already, that this LPS from Coxiella Burnetti is highly heterogeneous and a high molecular weight for all specific chain, the long chains. We have regions with high content viranose here. They are eluted first chromosome, and that's why, because of this heterogeneity, to solve the complete structure of this Burnetti LPS is very difficult. But nevertheless, we're able to do some basic studies on this LPS, and we were able to show this viranose and dihydroxystreptose and also mannose, the internal position of LPS or specific chain, and therefore they are better accessible to antibodies and so on. That's why they react there. So we also found that some of this viranose could be bound for in, and we think that we might have some homopolymer of viranose in this LPS1, but we are still not very sure. These are other structural linkages, but these are not here, not of too much importance. And we did a lot of work because in the early paper they was published in '87. This disaccharide galactosamine guanine acid 1,6-glucosamine should be present in all specific chain, but we were not able to confirm it. We spent a lot of time on this to find these two sugars in all specific chain. We should form actually backbone of this all chain, but this is probably not true. So that's at the end what's the tentative structure of Coxiella Burnetti LPS from UN phase 1. So we know on this lipid A, we know structure, I'll show you later. We know this part of inner core region because it's LPS2. We have some information on this outer core, and we have not find information on the structure of this all-specific chain. We know that viranose and streptose, those two sugars are located here in all-specific chain. Mannose is there, and then glucosamine and some minor amount of N-acetyl-galactosamine and glucose. That's it what we know on this LPS, but we work very hard on it and we want to finish this work very soon. But as I said, this viranose and dihydroxystreptose is very important for diagnostic purposes. And some years ago we developed a monoclonal antibody that is highly specific for viranose. And the primary paper has been published on it. And we tested this monoclonal antibody in ELISA, and we took some strains isolated, indicated, and all that were in phase 1 cells, and also LPS in phase 1, all of them contain viranose, and so actually they were positive in ELISA. But when we took 9-mal-2, which is truncated without 9-mal, it's virulent cells, phase 2 Priscilla 2, crazy, we know doesn't contain viranose, Henselin 2 cells, they were negative. And the same story was the cells, but LPS. When we did also some cross reaction with other bacteria like Salmonella, SR from abortus, and so we tested already much more of those. And also we tested it with Chlamydia psittaci, Chlamydia abortus. They were negative. That's very important also for chlamydia because most of antigens that were used for detection of serological diagnostics of Q fever, they sometimes cross-reacted with chlamydia. And again, the same story was with immunoblood, so I'm not going to repeat this. This was the same. Actually, ELISA or minimum of the world was confirmed by immunoblood. And now what we develop further this work, and we use this monoclonal antibody in a lateral flow assay to detect very quickly Coxiella Burnetti. That's very easy. You just take the suspension, you add here on this sample about 100 microliters of solution, and then you see if you got two bands here, then the reaction is positive. So actually we did it with Coxiella Burnetti isolate fluorian, this L35. Here's handselling, so it's positive here. So it's very easy. You just take it and drop 100 microliters. In 3-5 minutes, you see already result. And so it's very nice, very rapid for rapid detection of Coxiella Burnetti. So actually, we did again this. Here you have an imide. There is a 493. There's a phase 1. Phase 1, virulent phase 1, Coxiella Burnetti, you see it's a positive. Here you see phase 2 Coxiella, it's a negative. And here you see it's present on the virus, also negative. So it's very rapid in 5-minute detection. And here again is chlamydia, so it's an example. Here was negative is chlamydia abortus and chlamydia cytocin. And here again, so with some rickettsia, there's rickettsia rickettsii, there's rickettsia conori, rickettsia sibirica, and rickettsia kaplan, so they were negative. So that's rapid. Now we tested it with I'm saying it's the cell suspension of these microbes, but now we are trying to do this research also with environmental samples, and we will see what is going to be correlation on this. So, but this is a very rapid test. It might help for early detection in that for veterinarians. It would be very useful to take samples from abortion fluids or from feces of infected animals, so you might have results very easy. And this is a biosynthesis of Coxiella virulones here. It's very complicated. I don't know whether I should go through. Shall I? It's very complicated here. So that was a very actually schematic presentation of how this virulones is biosynthesized. So there's a lot of steps here. Some of those steps here at the beginning, they are already complemented, they were confirmed here. So I'll go very briefly through. Here is starts from fructose 6-phosphate. It's phosphomanose. It's isomerase. It's changed to mannose 6-phosphate, and it's mutase changed to mannose monophosphate. Then it's activated sugar. Activated sugar of mannose is prepared, and then you have it's mannose dihydratase. It's keto sugars formed here, but there was this biosynthetic pathway was proposed on the basis of bioinformatic approach, and some steps. These enzymes here, were all of them were confirmed here. Here we are able to show that this pyrosamine, what was actually given by, it should appear through bioinformatic approach. It's not possible because we have never seen pyrosamine in our LPS population, so this answer is not correct. Then there is a change to fructose here fructose, GDP fructose, beta fructose, then it's again this transformation here, and you end up here with a virulos. But here those steps are still it's just done by bioinformatic approaches. They are not very far up now. Now I will switch from these sugars to lipidae, and again it's a question why to study lipidae or gram-negative bacteria. As I said already, these are principal endotoxic components, and they play a major role in pathogenesis of bacterial infections. They are important contributors to massive inflammation, sepsis, and so on in gram-negative bacteria. They also promote activation of innate immune system by induction of common cytokines released by human cells. A lot of work with Coxiella Burnetti was done here in this laboratory here in Marseille, and there was also interest to what the structure-function relations on this. But we started in the past to study lipidae from LPS I and LPS II, and we did this through chemical component instruction, normality, tough-done electrospray MS techniques. And when we started this work, it was said this lipidae from Coxiella Burnetti is highly complex molecules, so it's very difficult to actually to detect the complete structure and to confirm the structure of these lipidae. But when we started it in 2004, we were able to show that this by far it's not complicated, but it's on the opposite, it's quite simple. And we were able to show that this lipidae has quite symmetrical structure and it has only 4 acyl chains. And 2 of them are amide-linked, hydroxylated fatty acids are amide-linked, and these non-hydroxylated were actually ester-linked. But in the same year, there was another paper from Zamboni, and he said, no, this our structure is okay, but should be like this. This is the same amide for lipid, amide for glucosamine 1 was the same. But here in this case, he said this esterified these hydroxylated fatty acids. But it is very difficult actually to confirm by mass spectrometric approaches. And we spent quite a lot of time on this. And we're able to show, again, it came out that actually, again, is such a symmetrical structure. It's a symmetrical structure. And maybe in this case, with this position, C4 prime position, could be this hydroxyl group, could be hydroxylated. As I mean, on this ester position here, could be hydroxylated with fatty acid. But this is not so easy. I would like to say this is not so easy to study these lipidae because these are highly heterogeneous. So you see here is a diphosphorylated maltodextrin. Here is monophosphorylated. Here you have masses for this structure shown here, but then minus CH2 14, you have another fatty acid. So its distribution of fatty acids are much more subtle. That's why those lipids are quite heterogeneous. You can have this one structure, but then you can have fatty acids with shorter chains or with longer chains. But nevertheless, we're able to show that those 2 structures are the main molecular species of this lipidae. But when you compare structure for this lipidae of Coxiella Burnetti with lipidae structures of highly endotoxic gram-negative bacteria, here is from Escherichia coli and the salmonella typhimurium. So you see at the first glance it was a huge difference because here you have 6 acyl chains, we have only 4. This is asymmetrical structure here, and we have a symmetrical structure. Length of fatty acids is also different. And it has been postulated that those lipids A and LPS, they contain the structure of lipidae structure like here in Coxiella, in here in Escherichia coli, salmonella typhimurium. These have a high endotoxic activity. And because of our lipidae from Coxiella Burnetti has a completely different structure, this should correspond to the fact that it has a low endotoxic activity. And actually, again, a lot of work has been done here, as Jean William and Eric Gigo, and they were able to show that really this endotoxic activity of Coxiella Burnetti and lipidae is much lower. But I'm only showing here one we did is production of DLF on human mononuclear cells, and we were able to show that we had, it's 100 times more lipidae should be used to trigger tumor necrosis factor activity compared to, this was from salmonella, which was that? Salmonella, I can read it very well. So you see, this is one of the proofs of the difference. So when we summarize it, it's a composition structure of two major molecular species of Coxiella Burnetti, lipidae and lipid are similar. And that was in the previous report, because as I said, these are highly cryptic molecules, they can change after phase creation. And those lipids are differ considerably from those published in other classical forms of enterobacterial lipids, and that these distinct structural features could be the reason for their enterotoxic potency. And this is another conclusion, these another suggestions here, that for this structural heterogeneity of this lipidae species is attributed to the action of latent enzyme that modifies the canolipid molecule. This has not been proved yet. And there is another assumption here that the variation of the lipidae domain serves as another strategy utilized by gram-negative bacteria to survive by providing resistance to components of the innate immunity system and helping to evade recognition mainly by toll-like receptor 4. So that's it. Thank you very much for your attention. These are Coworkers, they were working on this work. These are from our institute here, and this is from Germany. I did this, Dimitri was a lot of work on this lateral flow assay. This class Brandenburg from Germany did a lot of work on lipidae, and these are quotations here in Marseille. Thank you very much.
So, question? I will start. The virenose and hydroxystractose, they divide.
You have to talk.
Yeah, sorry. I'm talking about virenose and hydroxystractose. There are, do you know which enzyme in the bacteria? Do we know the enzyme in the bacteria which are encoding for this sugar, which are encoding to prepare the sugar, or it's a lipid? Do we know in Coxiella Burnetti this virenose and hydroxystractose? Do we know if the virenose and hydroxystractose are put on the LPS by the host in function of something released by the bacteria, so stimulation, a reverse factor which stimulates some enzyme which will put virenose and hydroxystractose by the host on the bacteria, or it's directly produced by the bacteria and put on the LPS.
Yeah, but there were actually, when we did the proteomics on Coxiella Burnetti, so actually we found genes that are encoding. I mean, it's deoxy, 3-symmetric transferase or sugars. We found this. They are put on this.
And are we able to take these genes and put in folate and to have LPS on the folate, for example, with the hydroxy and the virenose?
Yeah, but that's what I said, this last steps. I mean, on this biosynthetic pathway, I couldn't verify yet. That's hypothetical. Genes are there. Genes are there, but actually this, Jim Samuel did this work and I was helping him with verifying, because one thing is bioinformatic approach and the other thing is reality. And that there were many, you know, because we did a lot of work on, there will be some paper on this biosynthesis, another version, biosynthesis of virenose. Still,
the sugar will form or we don't form in the environment? Where the sugar, they are formed. You can find in the environment or not? the environment, those are bacteria.
No, they are only,
but those are organisms, something like that.
they are very unique. Some of them are, let's say, some of them the diet which is step 2, some of them which person is steptolized. But they will not be found in other.
And the example, it's just because they don't resolve the structure correctly, or because they have
because you don't see it on normal electrospray, you don't see it. When you run spectra, you can say it, because mass is the same. Mass is the same, whether it's there or there. But you have to cleave glucosamine chain or to do some modification of what we did to verify that this is a location like that.
And you know that now all these bacteria are traits. That is doing the bacteria directly on the plate, either plate, not doing any bacteria in cells or in eggs. Do you know if there is variation in the LPS? The LPS is still going in the way of virulent LPS,
that's a really good question. That's a really good question. Actually, what we really want to do, it could be because we have a mix. We have when we are isolating LPS, we have a small cell population, large cell population, we have mixture. And it would be very good what we plan to do to isolate from small cells and large cells to see what's different. Maybe it's different because maybe we have a mix. And there is another question, if you calculate, if you got the same LPS like in the case and calculating that's why we are doing now calculation on cells. But in principle, I cannot predict. But sugars must be there because because genes are there. The genes are there, and we confirmed those genes when we published first papers. We started proteomics very early, that's 2004 or 2005. We were able to show that those genes, and we were able to show and to detect proteins. Proteins are responsible for this passing.
And this system is LFR, lateral flow assay.
Yeah.
You try on patient?
No, but it's not used for, you can just use with Sarah. No, only when you took the cells.
Yeah, cells was
But this is important. This is important for veterinarians and for farmers, because farmers when they see abortion something like that, they just take it and it should work. We'll see. So that's application.
Nice question.
There was enough for them.
Okay, merci.
Thank you very much.